you know, they come and give you free massages and Reiki.
Oh,
And I
really?
took advantage of it for the first time last week because I was in such pain, and it was wonderful.
That's
She
awesome.
gave me an oncology massage and Reiki. And by
That's
the time
awesome.
I left, I had no pain.
Wow. What
It
else?
was great. I mean,
What
my
all does
pain
that
was so
include?
great. It was my blood work. She did an oncology massage, which is was basically just kind of laying of her hands and moving slowly. It's like a sissy massage, I called it. And then he is like the transference of energy. And her hands were very hot. It was
Bless you.
pretty cool.
Nice.
That's
It was
awesome.
pretty cool. I'll
Nice.
definitely do it again.
Oh, maybe we could have that at the summit.
Yes.
Ooh, chair
Have to send a house get on it
Maybe.
right
massages.
now.
Yes.
Oh, there he
Here's
is.
Gio.
Hi, baby.
Oh
Gio.
my
Hi,
goodness.
Gio.
Look how
Oh,
excited Caroline got.
look at how excited Carolyn's
Hi.
getting.
She did. She's like, ooh.
Oh,
Oh,
hi, Gio.
Oh,
oh,
hi, baby.
Oh,
Hi.
I love
Hi,
castle
Sarah.
babies.
Hi, guys.
Hi,
Sarah, you
Sarah.
look so
Hi,
pretty
hi,
tonight.
Sarah.
So many
Sarah's
babies.
here. Memo.
Yeah, there was something in the
Huh?
water, apparently.
I said you got the memo.
I got the memo. I'm here. It's nice to see everybody's
You're
beautiful
sick.
faces.
You're sick.
I'm not sick.
No, you sound like you have a sore throat.
No,
Maybe
not
This is
it's
called
really.
me.
diagnosing you online.
Wait a
It's
minute.
called a very little sleep, a lot of work.
Hey everyone.
Oh,
Hey.
David. Hi, David.
oh.
Hello.
Hello.
Look, we have
Francis, what's
babies
your little one's
tonight.
name?
Gio, baby Gio.
That's
Geo?
Gio.
Gio. It's Gio.
Nice. Love it.
Yankee, I love your
Dilemma!
Mine
Oh my
here
hair.
too.
gosh!
Oh.
Hi!
Hi.
So Gio and Caroline are girlfriend and boyfriend.
I
We've already hooked them up.
love it.
We're gonna
They're
get
gonna
that
get
Castleman
married.
wedding.
They're going to be at Summit.
They're gonna get married.
At Summit in 25 years.
I love
Yes.
Can you
It'll be our
imagine?
first castle wedding.
it.
We love it.
You can officiate, Dave.
You know what? I'd be honored, but I need you to be my partner, Penny, to help me with officiating. So we're gonna need to do it together.
Absolutely. Okay.
I think Penny's gonna be the singer. Wheels on the bus.
The wheels on the bus. There we go.
Yes, we've done
I'll
that
be the
one.
pre-ceremony entertainment.
Oh Lord.
I think you'll be the ceremonial entertainment also, Penny. I feel like you're gonna just be the entertainment.
And we can have a TED talk in the middle of the ceremony.
Pull out a PowerPoint and whiteboard. Sounds like a great wedding.
Yeah.
All right. I think it's a good time to get started. How's everyone doing today? It looks like we have a couple more jumping in. All right. So we'll start with my screen. Welcome, everybody. I'm so excited to have you here for another community gathering. Very excited to be able to get together just a couple short days before Rare Disease Day. So we will just jump right into that. Rare Disease Day is coming up on February 28th. And I have a couple of fun ways that you can get involved too. You can wear your stripes. If you didn't know, Rare Disease Day is all like zebra themed or purple, but you can also wear your CDCN swag, and that way you can celebrate what Castleman disease is as it's a rare disease and spread the word. You can tell everybody about rare diseases. You can share spread the word about the CDCN and what we do. If you know other Castleman disease patients, you can talk to them and let them know that we're here, we exist, and we're ready to support everybody, every patient. Even outside of Castleman disease, we have connections and resources that we'd love to help there. And you can contribute to research. We've talked to you guys a million times, but I'll say it again about donating blood or your medical records, the Accelerate registry. Any way that you can work with Bridget if you've been in contact with her already, it makes such a big difference in the research that we're able to do. And then, of course, show your Castleman Warrior Flex and then hashtag us on Instagram. I have a couple examples here on the right, but if you put hashtag Castleman Warrior Flex or at Cure Castleman, we'll be able to find your post and then share it. And I just have a ton of other examples. There's a lot of examples here of David with so many people, but all of the team as well, or other patients of just getting together and flexing and letting everybody know how proud we are to be Castleman Warriors and how strong we are. It's just such a fun like idea and movement to keep the momentum going. I also wanted to share that our Cooking with My Castle Fam cookbook is out. It's live for purchase, so you can scan this little QR code here on the right to get your copy. I have an example recipe here that was submitted by Francis. They're all recipes that were submitted by our community. They're personalized to the Castleman disease group. We have breakfast, lunch, dinner, appetizers, soups, salads, desserts, anything you can think of, treatment day quickies. All of this is right in this little cookbook. And then all of the proceeds that are raised from this are going to the CDCN. So we're so excited to have the warriors come together to put this together and help raise funds. I know I have Lisa and Penny on this call who have been really leading the charge on promoting this and selling it. Have you guys made any cool recipes out of here yet?
Really good. Anything I've tried is good. I know a couple people have tried Francis's Filipino chicken adobo and have said it's delicious.
Yeah.
I'm excited to try more of the international type recipes.
Yeah, it's cool. We have a good mix in here of like recipes from across the world. So many people have submitted. I think we had over 100 recipe submissions, which is really awesome. It's just such a like full book of recipes. It's awesome. All right. Then I also wanted to let you guys know we've been updating our physician referral list. We did an update in November. I've listened to some of the feedback that I've gotten since, and some more people have submitted new contacts. So we have an updated physician referral list. We're trying to make sure that it's easier for every patient to get access to expert care. So if you haven't received this yet, please email me at madison@thecdcn.org, and I will be able to answer all questions and send you a copy of this. It's really, really great. We've split in all of the physicians we've split them up by a category. So it's easy for you to know whether this is a center of excellence, whether it's a partner institution. So that would be people that might be on our scientific advisory board or have been like in collaboration with the CDCN for a couple of years, and they just really understand Castleman disease. And then we have these community physicians who are all people who have at least seen anywhere from like 2 to 5 cases of Castleman disease, and they're experienced in treating it. So they've self-selected that they want to see other patients. So this is just really helping to make sure that any patient out there can find someone closer to them. To further build on that, we're also creating a physician locator page. This should go live next week on our website. It would be split out there's 3 different types of icons on the map. But if you click on one, I have an example here for Ariella Noy. She's in New York. She's what we would consider a center of excellence because she's just so experienced with treating Castleman disease. She's involved in like multiple different programs and initiatives within the CDCN, and she really knows her stuff. So she would have that little star here, but that helps everybody find like here's the address of where she's her practices, the institution that she belongs to, and then you can click on a link. It will take you directly to that physician's main like profile page so that you can book an appointment through that link. But if you're looking for any additional contact information, of course, you can still email me, and I will give you that physical copy that has the more like robust contact information, like maybe their email address or their phone number, so you don't have to go through their contact page if you wouldn't like to. And then if either of these don't work, I've said this before, but I just want to make sure everyone knows we have the Castleman disease expert panel now. This is a really great service for your physicians or any of your providers, really. It doesn't have to be your oncologist or your hematologist. It could be anyone who's taking care of you're managing your care and your treatment. But the idea is that they could come and they can attend this virtual meeting so that they can get more information from other experts that are like familiar. It would be somebody like Ariella or Dr. Brandstatter, Dr. Van Rees on the panel. All of these physicians come together once a month to help give guidance to your physician so that they can help like further your care better and help you find like the right treatment for you. It's a really great program so far. Since we launched a year ago, we've talked to 40 different physicians and they're all spread across the globe. So that's a really great benefit of it is like, if there's not a physician near you that you can go see, you can ask your physician to come attend one of these meetings and it will help them treat you better. So it kind of like bridges that care gap that we might not have that you might not have been able to like find a physician or an expert near you before. All right, so what I think most of us are excited about is the summit is coming up this year. But this year we're doing 2 summits. So we'll be doing an East Coast and a West Coast summit. The East Coast summit is going to be your standard summit that you're pretty used to. It's going to be July 30th through August 1st. It'll be located in Philadelphia at the study. It's going to be pretty much what you're used to seeing. We'll have a warrior welcome, a full day of programming, and then a community fun day. And then this year, for the first time ever, we're going to travel and we're going to go to the West Coast for a summit too. So the dates of that are definitely going to be October 11th through 12th. We're looking right now between a couple different locations. It's largely between Las Vegas or Arizona right now, but it's just all like space dependent, of course. But we're really excited to expand that so that more people can attend the summit, whether it's like these dates work better for you, this location works better for you. But we're really excited to do it for a second time. I know this is a new concept though, so I just wanted to like give a second. Does anybody have any questions about this, or is there any questions I could answer?
Madison, I'm just going to make a quick comment. I'm super excited that we're all going to get together. And I think that is important because I saw some messages on social. We're not splitting up the patient summit. We're holding the summit in Philadelphia like we always do. We want to encourage everyone to come to that. And we're also adding on essentially a satellite summit, like an extra one. So the intention is not to like divide the thing that we all love so much. It's to have the thing we all love so much in July, like we always have it, and then to also have sort of a satellite, an extra summit that's happening that's going to be maybe more convenient for people that are on the West Coast. So I think about it less of an east and west, and less of like an either or, and more of we're doing the same thing we always do, and we're adding on a satellite summit.
Yeah, I think that's a really important point. You're welcome to attend both. It's not an either or option. We would love to have like the more the merrier for each one. We'll still be offering travel grants to go to the East Coast or the West Coast. What I will say is you can get a travel grant to go to one or the other, but you're still like welcome to attend both if you would like to. All right. Well, if there are no questions, I think it's a good time to move on to remind you guys that there's always the opportunity to contribute to research and always the opportunity to work with Bridget and the Accelerate team. If you are an IMCD patient who has either been pregnant after being diagnosed with MCD, or you've been diagnosed with multicentric Castleman disease as a child, we're always looking for new blood donations or medical record donations. It helps us do the work that we're able to do. So you can reach out to either this email address here or the phone number. I know that you guys are, I think pretty much everyone I saw on this call already is very familiar with this, so I won't spend too much time on it. And this is still the same thing. We're also looking for saliva donations. It's not just blood, so it'd be like a swab of your mouth. But all of this is really helping us to be able to do these like longitudinal like historical studies of like Castleman disease patients and better understand what Castleman disease is, the mechanisms behind it, and try to predict different like if we can come up with treatment options or other like diagnostic criteria to help like reduce that diagnostic odyssey for you guys. All right, so now I'm going to pass it over to Dorottya. She's online here with us. She is going to share a little bit about the research she's doing in the cabin 1 mice.
Hi, everyone can hear me?
Yep.
Yes.
Right, great. So should I share my screen or can I advance the
I'll advance it for you. Yeah. So
or maybe can I just share it because sometimes I want to point out.
Oh yeah, absolutely. Go ahead.
Yeah, maybe I just share it. That's okay. And
while Dorottya is pulling up, I'll just give Dorottya a quick intro that Dorottya did her MD PhD and did her training at Penn in pathology and has been really committed to Castleman disease for many years now and has been an awesome collaborator. She's now a young faculty member at Penn and just doing such great work. So excited that Dorottya is going to share.
so thank you so much, David, for the introduction, and thank you so much for the invitation. I'm very excited to be here and talk about my my research. I would like to apologize. It might be a little bit too technical at some parts, and also I didn't know that that there will be such a young audience as well. So anytime, just feel free to ask any questions, and I'm always happy to answer. So let's just start with the introduction, which, although I guess most of us know what IMCD is, but just a warm up, let's just say a few words about that. So IMCD is a very enigmatic and many times life-threatening condition that is characterized by episodic hyperinflammation. And also together with hypercytokinemia, meaning that there are increased inflammatory markers such as IL-6 or CXCL13 in the patient's blood samples. Also, these patients present with enlarged lymph nodes at multiple stations of the body, which display a characteristic lymph node histology. The etiology is so far is unknown. However, it is suspected that IL-6 and mTOR could be crucial players in the pathogenesis, as inhibition of this pathway resulted in significant clinical improvement improvement in a large set of patients. However, 1/3 of patients still do not respond to either IL-6 or mTOR inhibitor, suggesting that there's a dire need to identify additional pathways that could contribute to IMCD. So the diagnosis of IMCD is pretty difficult because it requires the correlation of pathology, laboratory data, and clinical findings. However, the pathology diagnosis is the mainstay of the diagnosis, as abnormal lymph node finding is the major diagnostic criteria for IMCD. These lymph node findings include abnormal germinal center formation, increased stromal elements, increased blood vessels, and/or plasma cells. But usually these features are on a spectrum. Sometimes we get more or less of each features. And among these four, I would like to highlight the abnormal germinal center formation because this will be important in later part of my talk. But first, let's just talk a little bit about what a germinal center is. So germinal centers are specialized structures within the lymph node, and it can be divided into several parts, such as the mantle zone, which has the naive B cells, the dark zone, where the B cells proliferate, and maybe you don't see it because, and also it has a so-called light zone, where the B cells can migrate after the proliferation, and they become a fully differentiated plasma cells that secrete antibodies, and that these antibodies will help to fight against infections. And basically, this process helps the body to make stronger antibody response over time, build long-term immune memory, and avoid any weak or ineffective responses. Oops. So under normal condition, as a pathologist, if I look at the lymph node specimen, this is the picture, what I would like to see. This is a normal looking germinal center with this darker circle around it, which we call the mantle zone. And it has a nicely polarized germinal centers. That basically means that we can discern a dark zone and light zone. However, when it comes to IMCD, we get a completely different picture. So on the right side, you can see an example of an IMCD lymph node, which has like a much smaller germinal center, which is highlighted with blue. And also, as I mentioned, they have a prominent vascularization that many times penetrate into the germinal centers, causing the so-called lollipop sign. And they have a much more prominent mantle zone that we call onion skinning. So as I already mentioned at the beginning, the etiology of IMCD is so far unknown. Over the years, several hypotheses have emerged, such as a presence of a pathogenic infection, an autoimmune disease, or on a potential acquired somatic mutation. However, there is so far there is no solid proof for either of these. And after that, we became particularly interested in a potential genetic basis of immune dysregulation. And in order to prove that, we decided to sequence one of the IMCD patients and their parents. And this very detailed analysis identified 2 mutations within the gene called cabin1. And based on the analysis, this was predicted to impact the protein function, and this is why it was suspected that it could play a potential causal role. But why we became interested, but why cabin1? Why did we care about it at all? So in order to understand, first we need to see what cabin1 does for a living. So cabin1 is a very important orchestrator of an immune response through 2 main targets. One of them is the inhibition of a molecule called calcineurin. And calcineurin is very important in T cell activation regulation and also T cell differentiation. And there is another target called a transcription factor called MEF2D, which is also important in T cell activation and T cell differentiation, and also very important to establish a connection between T cells and B cells. That is a critical step in germinal center formation. And the reason I'm mentioning this because I would like to talk about this a little bit more in the later part of my presentation. So after identifying these mutations and knowing what cabin-1 does, our main hypothesis was that cabin-1 mutation could contribute to IMCD through abnormal T cell activation. So now a few words about the current research in IMCD. So until today, the research into IMCD is mostly relying on analysis of patient samples, which is crucially important and gives us many important information. However, when we really want to look at the basic mechanistic studies, the fact that it's a pretty rare disease, and also the fact that many times when this patient comes to our site, had been already treated with multiple immunomodulatory drugs, could hamper the interpretation when we really want to look into deep mechanistic things. So this is what an animal model can overcome, because with an animal model, we can look at basic mechanistic pathway, and we can also use a very high number of them. So, however, unfortunately, in IMCD, so far an animal model that could answer key questions has not been available yet. So because of that, we decided to introduce the previously identified cabin-1 mutation into a mouse model using a gene editing technique called CRISPR-Cas9. And with that, we generated 3 different mouse lines. One of them has 2 copies of one of the identified mutation, the V2185M, another one with 2 copies of the other mutation, L816F, and also a so-called compound heterozygous mice, meaning that it had both mutations, and basically that was the mouse that mimicked the original patient. And first, what we found that these mice were completely normal. They had normal weight and size and had compared to the wild type control. And basically they had no striking difference in their populations. So after seeing that, we were really ready to give up this project because we thought that cabin-1 might doesn't do anything. But then it just crossed our mind that maybe a second hit might be necessary to see any phenotype. So that's why we made the decision to infect these mice with a mouse-specific virus called lymphocytic chorea meningitis virus to see what happens. And after infecting these mice with this virus, finally we could see some difference. On the left side, you can see a survival curve, and on the right side, you can see a clinical severity score, which are basically complementary data to each other. So on the left side, you can see that the mice that are highlighted with red and green showed increased mortality, meaning that many more mice died after the LCMV infection, and this was reflected in the clinical severity score. And if we take a closer look, the most severe mouse deaths happened in the mouse line, which had 2 copies of this 22185M mutation. And the green line represented the compound heterozygous mouse group, which basically was identical to the IMCD patient. So, but since we saw the most robust response in the homozygous V2185M mice, we decided to continue the experiments using this mouse group. And more importantly, one of the most important findings of these studies that we found that after infecting the mice with this virus, they showed a very similar cytokine profile that we see during human IMCD flares, with increase in IL-6, VEGF-A, MCP-1, and CXCL13. And among these 4 cytokines, I really would like to highlight the marked increase in CXCL13. And we were particularly interested in this result for several reasons. For one of the reasons why we were happy about it was the paper that was published by David's lab a couple of years ago that basically highlighted that CXCL13 was the top upregulated protein in IMCD, and it was speculated that it must play a very important role in the pathogenesis. So the other reason, the more mechanistic reason why we became interested about this result, so to understand that, we need to understand what CXCL13 does. So CXCL13 is secreted by non-hematopoietic cells, and then CXCL13 exerts its effect through its receptor, which is CXCR5, which can be found in abundant number in B cells and also on a specialized T cell population called T follicular helper cells. And these T follicular helper cells play a crucial role in germinal center formation and maintenance. And as I mentioned in the beginning, this is one of the hallmark finding in IMCD. So we thought that might be a clue that we may need to investigate this pathway a little bit more. So in this cartoon, I just wanted to kind of summarize how a T follicular helper cell can contribute to germinal center formation. So they are from naive CD4 T cells that eventually migrate to the germinal centers. And within the germinal centers, secreting different molecules, they help B cells to become germinal center B cells. And this whole process helps them to eventually differentiate into fully formed plasma cells. And plasma cells are the cell types which are secreting the antibody that will protect us from from different pathogens, viruses. And if this T follicular helper cell is not working well, this process just cannot happen. So since the LCMV totally disrupted the lymph node architecture, we had to use a different experimental approach. So we used like a model antigen, which was an ovalbumin mRNA LNP. So basically, it has a similar concept that a COVID vaccine is, which basically also uses mRNA LNP. But this time we had a molecule that encoded a model antigen called ovalbumin. And this molecule is known to induce a very robust germinal center response. So after immunizing the mice, we collected their lymph node and looked at the histology and also the different immune cell subtypes by flow cytometry. And one of the most striking results that we found that after this immunization, on the left side, you can see the control mice lymph node, and they had a very nice looking germinal centers, very similar to the one that I showed you at the beginning of my presentation. However, in the cabin-1 mutant mice, these germinal centers were much smaller, and this darker part, the mantle, was more prominent. So which kind of was very similar that what we would expect in IMCD as well. And this was reflected in the numbers as well as the number of germinal center and the size of the germinal center. So just a little mechanistic background, as I talked about the CXCR5 positive T follicular helper cells, then we became interested if we see any difference in this immune cell population as well. And because of the small germinal centers, we totally expected that they will be decreased. However, we got the complete opposite. We found that actually this population was increased in the cabin-1 mutant mice compared to the control group. So this was something that we found that could be mechanistically interesting, that why we see more T follicular helper cells and decreased germinal center reaction. And although the real answer is still underway, and we are keep doing experiments to prove this point. But as we were looking into the mechanistic aspects, we found this paper that was published in Science Immunology that basically described that the MAF2D, one of the target of cabin-1, is responsible for an expression of a protein called SAP that is very important in the T-cell B-cell connection. That is basically one of the most important part of the germinal center formation. So because of the strong connection between cabin-1 and MAF2D, we decided to look at MAF2D levels in our cabin-1 mutant mice. And so for those who are not familiar with that, this is a Western blot. And basically, just to read it, what we did, we isolated T cells from control mice and also from cabin-1 mutant mice, and we stimulated them. And these numbers are just showing the different time points we stimulated them. And under the blue column, you can see the control, and under the red column, you can see the cabin-1 mutant T cell response. And basically, what you really have to look at if the band is darker or not compared to the wild type. And you can see that in the cabin-1 mutant mice, there was much more MAF2D in the cabin-1 mutant mice compared to the control, both at baseline and after different time points of the stimulation. So because of the sake of time, I cannot really talk about more experiments that we have done so far, but we basically think that this could be a key mechanism why we see this disconnect between the CXCR5 positive T follicular helper cells and the small germinal centers, that we think that because of this cabin-1 dysregulation, MAF2D inhibition is decreased, that basically decreases the expression of this protein called SAP. And because of that, the T cells and the B cells are not able to connect properly. And this is responsible for the small germinal center response. And this is just a human data that seem to kind of, that's kind of very promising and maybe justifies what we are thinking. So on the left side, you can see a control germinal center, which has a lot of red and magenta, which corresponds to the T follicular helper cells. And you can see in IMCD, this red and magenta, there is really not too much of it, as you can see it here, suggesting that maybe there is really a migration problem that prevents the T cells to connect to the B cells. So just a summary and some take-home message. So what I just wanted to tell with my result, that with all these technical details, and I apologize, it was like too detailed, that this experiment was basically the first demonstration that a genetic mutation could predispose to IMCD-like features. And we saw that the cabin-1 mutant mice showed small germinal centers and decreased germinal center response, similarly that we see in the human disease. Mechanistically, interestingly found on kind of paradox CXCR5 positive TFH-like differentiation. And more mechanistically, we saw evidence of dysregulated MAF2D pathway. But all in all, what we really hypothesized with our mouse model, that this could be used as a new platform to better study IMCD, uncovering potentially new biomarkers and targets for therapy. And here I would like to thank my mentor, of course, David, who has been a great help to me all the way through. And also I have another mentor, Taku Kambayashi, and I also spend a lot of time in his lab doing these mice experiments. And of course, thank you very much for both the CDCN and the Kambayashi lab. And thank you so much for letting me talking here. And happy to take any questions.
Awesome job, Dorottya. Thanks so much for presenting. Maybe one question that I have to kick it off while other people are thinking through questions. Any thoughts on implications for treatments for patients based on this? Any drugs that come to mind based on this concept of T-cell hyperactivation?
Oh, generally, if there is really T-cell hyperactivation, sirolimus, mTOR inhibitor is always a good thought. I just don't really, I think in the future, if we really have more proof that MAF2D is so important, we should see I don't know if there's any treatment that would target MAF2D, but look at any downstream of MAF2D or any pathways it affects. So I think that could be something that we could consider like trying.
I agree. The other one that comes on for me is cyclosporin. If cabin one's a
Like a carcinoid,
negative
yeah.
regulator, maybe cyclosporin is another way to go. I
Oh
know that
yeah,
cyclosporin is used in Japan in some patients.
yeah. I mean, most probably generalized some immunomodulatory drugs could make a difference. But yeah, I mean, it would be nice to have some targeted. And if there would be anything that could target MAF2D, that would be awesome.
What questions do you guys have for Dorottya?
There's actually
I have a question.
Oh, go ahead.
Thanks, Madison. Should we be getting CXCL13 measured during,
Yeah.
early diagnosis or even during our regular blood work?
Yeah, I think this is something that was actually, so I work in the pathology department, and actually this is something that was already discussed at one of the meetings the other day. And so since we think that CXCR13 could be so important, yeah, I think maybe that could be something to incorporate it into the the standard inflammatory panel. As far as I know, it's not. I'm not sure if it contains right now. It's mostly like IL-6, IL-2 receptor. Yeah.
Yeah, that's right. So it's not currently available, Andrew. We looked into seeing if we could set one up at Penn, and it's been difficult so far. So it's not currently available. We really recommend that if you're going to do sort of exotic labs that aren't typical, we recommend VEGF and soluble IL-2 receptor. Those are two labs that are not as widely available. I can put them in the chat. But those seem to be pretty good at helping to pick up an early sign of relapse and also track how you're doing.
Yeah, but
Okay,
I
thank
know
you.
that it's been also been an active discussion at Pan, at least if we should. It's more like it's not. I think it's not really the usefulness. It's sometimes if there is a really reliable assay that could be used.
I had a question. Thank you for the presentation, first of all. I guess, sorry, what in the actual patient data itself, what's kind of the prevalence for this cabin-1, somatic mutation?
So this specific mutation, so far I don't think that that was the only patient with this specific mutation. It's more like, so we really think about this cabin-1 mutation could be like a proof of concept that could help us to identify like common pathways that IMCD shares. So it's not like you need to have cabin-1 mutation to have IMCD. It's more like it could maybe shed light what are those pathways that are affected and give IMCD like phenotype. And I think that's why this study can be important, because if we eventually identify like a bigger pathway that is affected, that could be maybe another drug that we could use, and maybe that 1/3 who doesn't respond could benefit from that.
Thank you.
I have a question. Does a newly diagnosed patient who's UCD ever titrate up to IMCD or the new subtype?
Oh yeah, I think that's a good question. Especially like, so it's always hard to tell if something is like localized or not. So I think if it's a real UCD, it's more like, especially if it's like a hyaline vascular, we look at it more like a follicular dendritic cell, like pre-neoplasm. So, but when the patient have like some inflammatory symptoms as well, but we only see like one lymph node. That's kind of always hard to tell if it's like real localized disease or just kind of missing something. So yeah, I think I'm just not, but answering the question, I'm not aware of a specific patient who kind of upgraded to IMCD later on.
Yeah, and I'd say the same thing. There have been no cases that we're aware of someone who started out with UCD and then developed MCD. That's never happened that we're aware of. We think about them as different diseases as opposed to sort of like a spectrum of the same disease.
Yeah, I think somebody raised
Yes,
their
Stacy.
virtual hand up.
I see Stacy's got her hand raised. I don't know if that's scary.
Hey, hi, it's Gary. So just to clarify, it sounds like with the mutation, you only detect the Castleman similarity or tie-in after some sort of like a viral or infectious stimulation.
Yeah, exactly. That's the case. So, that I don't. So it's yeah, that's something that maybe we can consider. That of course we don't know yet, but could help us wonder if patients who present with IMCD, maybe they had some kind of viral infection or something that they are not aware of that eventually triggered the disease.
Thank you.
There's also a question in the chat that says, does this suggest that a virus triggers the disease?
Yeah, I mean, we don't know yet because there was always kind of in terms of the etiology, of course, a potential infection was a possibility, but there was no specific virus that really triggered that anybody found that could trigger IMCD. But it's not unreasonable to think that maybe some kind of latent infection or something happens that eventually triggers the first IMCD flare. And also, I would like to mention that as a pathologist, it was all during the COVID. It was sometimes a little bit difficult we got lymph node when it was hard to distinguish if we saw like IMCD like picture because in the setting of COVID or it was a separate IMCD. So I mean, it can, like for example, a COVID infection can also give a cytokine storm like presentation. So I think it's a kind of fine line.
Esther?
Is there any benefit for us to get genetic testing?
So I think for even if there is like no specific mutation that we are aware of that could 100% justify IMCD, I think it's still a good idea if there is a chance to do genetic, because I feel that the more data we will have, the more chance that we can find, what could be common in these patients.
Yeah, I just jump in to say I totally agree, Dorottya. The value for doing genetic testing for yourself right now would be to see if there's genetic change that's known for another disease, and that it actually would help you figure out you have something other than Castleman's. Because to Dorottya's point,
yeah,
you might find that there's some other genetic mutation. Because right now we don't have a clear genetic driver in Castleman's, but as you know, I think at this stage it's maybe over 200 patients, maybe closer to 300, have given DNA samples for us to do a very large genetic study. Mike Gonzalez is currently doing the analysis right now to look for genetic changes that could be potentially involved in Castleman disease. And so hopefully from that we'll identify a few genes that really could be useful. And the other thing I'll mention is that when we talk about genetic sequencing and genetic changes, You can have you can have mutations that you inherit, which are present in every cell in your body. Those are what we call germline mutations, but you can also have what are called somatic mutations, where the mutation is only present in one part of your body. And so, if you do genetic testing of, let's say, your saliva, you'll only figure out the mutations that are present everywhere. You wouldn't figure out if you have, let's say, a mutation in one of your lymph nodes, for example. So it's not just whether to do the sequencing, is also what part of your body is getting sequenced, which makes this all really complicated.
Right.
Are there any other questions out there? It doesn't have to be just about this. We can open up the floor for any questions, really, whether it's about the summit, your personal experiences, whether it's about the research that Dorotya's sharing, whatever questions you guys have, now's your time.
I have another question. So last month I think I got a viral infection, just a common cold, I think. But after it was done, I started to flare up with my IMCD type rash that I had when I initially, you when I first got symptoms of IMCD. And I was wondering if that's common amongst this group to, when your immune system is flared up, that an MCD relapse or MCD symptoms start to show. Before that, I've been perfectly fine until I got that infection.
I really don't. I teach differently. These competitive answers, I'm not.
Sorry, just muted that real quick. I think you're good to keep going.
Oh yeah, that was my question. I was wondering if anyone else has had MCD symptoms coincide with like a cold or a flu or something like that.
What's the subtype you have, Andrew?
IMCD, specifically IMCD IPL.
I have oligocentric, and when I get sick, I tend to have a few symptoms come back mildly.
I find I get night sweats for several days to a week or so after I get over any kind of a cold or flu.
So I was wondering, when you were giving that research talk, I was wondering if, infecting the mice with a virus can trigger that type of IMCD response.
Yeah, I think it's totally possible based on what I mean. Of course, this is just a mouse, but basically, although we are not aware of any specific virus, but maybe like any immune stimulation in general could predispose potentially.
Okay, thanks.
Hi, I also would like to share, you're talking here for Brazil. This is the patient, I'm the translator, but I would like to share how she started with the symptoms. She didn't start as having a cold or something like this, but it was confused, it was misdiagnosed with Raynaud's syndrome. So they thought that it was about the lymphomes, but and then they figured out because of the acids that was something else because she has a lot of liquid accumulated in her belly. So it wasn't associated with a cold or anything, but it started with this pain in her belly with the accumulation of liquid. And she was almost diagnosed with lymphoma, but now she was diagnosed with this disease. Thanks.
Yeah, so sorry to hear about what you're going through. I saw well, you put in the chat, and it sounds like you've gone through so much with so much ascites and so much fluid that's been taken out of your belly. I'm so sorry to hear about that. I guess a question around have you guys tried siltuximab, or what treatment has been tried to get you into remission?
Yes, is the one that you said, the C2.
Siltuximab?
Yeah, that one.
And how long has it been? Yeah.
That's not
Wow.
fair. 7 months.
7 months of siltuximab? And
Yeah.
still all that
This,
fluid? It might be that other treatments needed on top of the siltuximab. It might, I mean, that's so much fluid for so long. It might be that
yeah.
you need to add rituximab, you might need to add sirolimus. If there's a lot of other symptoms, you might even need to think about chemotherapy like Cytoxan.
Chemotherapy, you said?
Yeah, possibly chemotherapy. I mean, at this stage, 7 months I mean, that's serious liver dysfunction to have that much fluid in your belly. It might be that other treatments are needed on top of the siltuximab.
Thank you. I was translated to them.
Yeah.
Yeah, their doctor will participate in the next conference on the
Oh,
13th.
great. Oh, great. So you're
Yeah.
tied in with Madison. Great. And I shared in the chat just for everyone on what Madison set up with this expert panel is so
Yeah.
wonderful. Patients, like
Yeah.
yourself and others getting access to these experts. So hopefully
Yeah.
they'll be able to guide you to some treatment to help you. I'm so sorry all you're going through.
Thank you so much. So that's the recommendation. So keep with the medication that you said, plus the chemotherapy.
Well, no, I wouldn't say that that's a recommendation. I think I'd say that I recommend that you talk to the doctor about adding additional therapies on, determining what it is. Rituximab might make sense, sirolimus might make sense, but yes, I think the point is that 7 months of siltuximab and that much fluid in the belly suggests that some other drugs might be needed.
Okay. Thank you. Any
Hi, my name's Paul. I'm in the UK. My son is the young man that's had IMCD Tafro. He was 16 when he came down with it. We were in hospital for 5.5 months. He's now a year in remission, so everything's going fantastically. Shout out to the guys at UCLH; they were brilliant. But he's now going back for every 6 months for some tests. But all we're doing is basic blood tests. Is there anything that we should be looking for, or any other markers to check on a regular, say, 6 monthly basis of things to monitor?
Yeah, Paul, the two things that I'd suggest that aren't typically measured are VEGF and soluble-2 receptor. I'll put them in here. VEGF, soluble-2 receptor. Those are not typically measured, but those would be two tests that they can help to give you a sense before relapse that someone might be having a relapse, or have a sense for how calmed down is the Castleman disease, especially IMCD Tafro. I would measure those two. And then I think the other thing too is to encourage your son to really pay attention to his symptoms and how he's feeling. I know that a lot of the folks on this call, we have to be diligent, and if we feel more tired than usual, we need to know, is it just that we've got some cold, or is it that there's a relapse of the Castleman? So really being on top of his symptoms and how he's feeling is really important.
Unfortunately, he's now 17, coming on 18, and thinks he's indestructible. So, but yeah, every now and again he'll faint, collapse, that sort of thing. But on the whole, he's just doing his thing now again. So, but yeah, we're trying to do that. It's not easy.
Well, Paul, I'm so happy to hear he's doing so well, and thanks for calling in at midnight or later your time.
Yep, 1 a.m.
Oh, geez. Any other questions from the crew?
David, when I had my original presentation, I had my fluid on my belly for a good 12 months before it was drained. And then it was only after my blood test results was showing that there was improvement. Was drained over 24 hours, and I had over 4 gallons of fluid on my belly.
Wow.
It was very heavy.
Goodness.
So that was how my doctor treated me, and I'm fine now. I still every now and again feel like there's ascites on my belly, but nothing like that.
Wow. Gosh, I'm so sorry to hear that. Yeah, I mean, ascites is just a sign that your liver and usually your kidneys as well are just not functioning properly. And usually if you can get the right treatment to control the Castlemans, the ascites will go away even without needing to get it drained. But sometimes, like maybe in your case, Allison, the treatment wasn't having an effect fast enough and you had to get it all drained off.
Yeah, I was the biggest loser that week, losing over 40 kilos, which is about 25 pounds or something like that overnight. So
more.
it was like, woohoo!
Wow. Amazing. Oh my gosh.
I felt a lot of relief after that drain.
Wow. Incredible. Well, I think it's about time to wrap up. But before we wrap, I did want to call. I don't know if Madison told you, she's on vacation this week. Madison is off. She's on vacation. She's not supposed to be working, but it's Rare Disease Week, and so she's coming out of her vacation to host this call. So big shout out to Madison for making the time during her vacation to join on with all of us.
Oh my gosh, I love
Thanks,
being here
Madison.
for you guys. No problem.
Are you somewhere warm? Because I notice you have some color.
I do have some sunburn, yes. I'm in St. Martin right now, which I know is hard to say when some of us are experiencing some extreme snow.
It's very cold in Philadelphia, Madison. We don't want to hear how much fun you're having.
None at all. It's terrible. But such a pleasure to be.
Well, thank you guys all so much for calling in and being a part of this. We've got the best community in the world and the best leaders with Madison and so many other amazing other people on the team. Penny, so good to see you. So good to see you guys.
You give the kids hugs.
I will. Good night.
All right,
All right,
everyone.
bye,
Happy Rare Disease Day.
Happy Rare Disease Day.
Rare Disease Day.
Bye, everyone.
Pode sair?